My dear viewers, you are my greatest inspiration. You have supported me and inspired me all these years, and I keep on making more videos. Dear viewers, please note that this video is rather compact, as a starting point, explicitly presenting the key definitions as precise and concise as possible, the way you should write in your exam answer papers. Elaborate elucidation or mathematical derivations are beyond the scope of this video. I have basically focused on the implications in the context of DNA double helix. I have over 200 videos on elaborate lessons in thermodynamics and physical chemistry in my channel that you can consult as per need. These are the chapters this video highlights. When heat is added to a system, it transforms into another form of energy. It either increases the internal energy and/or does external work. We rotate a turbine to convert the mechanical energy into electrical energy. Likewise, the electrical energy can further be converted into light or heat energy by lighting a bulb. Alright then, let's proceed with enthalpy change. So, this is what we get about enthalpy change. Enthalpy is 0 for elemental compounds. Enthalpy is non-zero for water, regardless of phase. Enthalpy of an ideal gas is independent of its pressure. The enthalpy change of a reaction is roughly equivalent to the amount of energy lost or gained during the reaction. When a substance changes states from a solid to a liquid to a gas, such as water going from ice to liquid water to water vapor, the atoms and molecules have more freedom to move. This increase in freedom corresponds to entropy. Now, I will briefly go through these key themes. As we all know, DNA molecule is made from subunits attached to each other in a long twisted ladder-like chain. Hydrogen bond formation takes place between complementary strands, while stacking interactions are formed between adjacent bases. The destabilizing repulsion between the negatively charged phosphate groups in the backbone of the DNA strands is overcome by the favorable hydrogen bonding and base stacking interactions. These processes are precisely orchestrated and coordinated. Whilst the pairing of conjugate bases is crucial for recognition of complementary strands, stacking of the flat apolar surfaces of the base pairs reinforces the DNA double helix formation. Two factors are mainly responsible for the stability of the DNA double helix: base pairing between complementary strands, and stacking between adjacent bases. Now, how to discuss the roles of enthalpy and entropy in the context of DNA double helix association-dissociation. Okay. Right. Now, what is the purpose of exploring thermodynamic aspects of the interplay of the forces stabilizing DNA double helix? Relative contributions of base pairing and stacking to DNA stability provide a fundamental conception of DNA structure as well as of significant implications in biological processes. The enthalpy associated with base stacking is favorable, while the entropy associated with base stacking is strongly unfavorable. We have to bear in mind that constructing the form of the helical arrangement of base stacking brings in an extremely ordered state. DNA base pairing is entropy-driven, and is coupled with the enthalpy-driven van der Waals interactions between the tightly packed apolar parts of the base pairs. The cooperation of entropy-driven base pairing with enthalpy-driven base stacking defines the efficiency of DNA double helix propagation. The hydrogen bonding between conjugate base pairs provides a purely entropic contribution of approximately 40% to the Gibbs free energy, while the enthalpic van der Waals interactions between the base pairs provide the remaining 60%. Whilst binding to the major groove is largely enthalpy-driven, minor groove binding is entropy-driven as a result of releasing the ordered water. Now, with reference to DNA association-dissociation, hydration of the apolar surfaces of bases become exposed along with disrupted hydrogen bonds upon duplex dissociation. Enthalpy results from dissociation of the stacked base pairs. The entropy is expected to be strongly negative because water molecules become ordered around the newly exposed polar groups. On the other hand, upon association, enthalpy of base pairing is negligibly small, while the entropic contribution is considerable.
Please remember to like, subscribe, share,